All right, good evening, everybody. Welcome to another video lecture. I hope you can hear me okay. I am going to do this biotechnology section of your DNA unit, and I'm going to talk about this in a few small parts. So I'm going to try to make this just a few minutes, and then I'm going to actually do a part two. Biotechnology is a really neat field, and again, let me start this here. You'll find this in your packet on the pages that I assigned you. Okay. And here we go. It's really interesting what biotechnology is. Biotechnology is technically defined as using living organisms to help humans do something. It means it that simply. Using other organisms to perform certain kinds of jobs or to make, usually it's making some things that we want. Usually it's medicines and things like that. The one that we're going to talk about of biotechnology is really using DNA technology. We aren't going to talk about really fancy computers and things like that. We're really going to talk about how DNA and the technology that DNA has, how that helps us make new things for humans. It's really fascinating. So you see this picture here at the bottom. That's a chromosome, and you can see that it's really wound up. And if you look over here on this side, you'll actually see that blue backbone, the sugar and the phosphate, and there's the bases there. And the truth is that DNA is not only wound up, as you know, like a ladder here, but it's also then wound around even more around these proteins that are called histones. And that's not anything that's a big deal to memorize, but remember that when we're talking about genetics, we're talking about a complete set of genetic material. That's what we call the human genome, the complete set of genetic material that a human organism would have. And for us, that's 23 pairs of chromosomes and lots of genes, about 25,000 genes. And again, all these chromosomes that we're talking about, the DNA that you've taken out of strawberries, that all can get stuffed into the nucleus. And it's because the DNA gets wound up even more around these pink things here called histones. These are these proteins. And then we call that a supercoil because it's coiled so tight. And that's an actual picture right here. So as you saw from the movie that I started to show you, from about 13 to 15 years there, it took them to actually sequence out the whole human genome, and that is called the Human Genome Project. And we have done that now. We know every gene that's on every chromosome, every code that codes for any trait that the human has. And that's 3 billion base pairs, that I think you know that already. And of course, that's all stored in databases and on a computer so that if you wanted to get yourself tested and find out your code, you could see if you're going to have a normal gene or something might be different about your gene. And sometimes you can diagnose and treat a disease if you know that you might be not keeping cholesterol real well in your body and that it's blocking up your arteries. You can learn that early and take medicine that will help you with your cholesterol and your fat or something like that. So we can actually, by knowing the base codes, we can actually fix a lot of problems. And we're going to talk a little bit more about this later, but as you saw in the movie, there's a lot of ethical problems with this, with knowing everybody's code and knowing if, like, what if a place you wanted to apply to for a job looked at your DNA and said, nope, sorry, you're going to have a heart attack maybe, so we know you're going to have heart troubles because it's in your code, so no, we don't want to hire you. And those are kind of ethical and legal issues that affect society. Remember that as we go through here, you can always stop this and kind of take some notes. Now, of course, all cells of all living things have DNA, and that can be read, and it can be read by lots of different cells. As you might remember, we have a lot of the same genes as bananas. So we can take different organisms, and as long as they're both made from, have DNA, which they are, then we can actually use them, and that's called manipulating DNA. And the type of technology, as I mentioned, is called DNA technology, and it's called recombinant. I want you to think of the word combine. What that means is that you can actually take genes from different organisms and put them together. You might say, why would you want to do this? Well, in biology, in medicine, especially bacteria are used a lot because they contain little short circles of DNA, little loops of DNA called plasmids. And I'll show you these on a picture. And these plasmids have have DNA. They're made out of DNA, and they're a loop. And you can actually break them, insert a gene into them, and then the bacteria multiplies. And when a bacteria multiplies, it multiplies much more quickly than humans do, and you can make many copies of that same gene. I'm going to show you that in a picture. But this is a really neat thing that you can do with bacteria. Where let's say you want to have more of a certain chemical made that you want to give to people to help them, you can actually insert that gene into bacteria, and then the bacteria split and split and split and multiply, and you make more and more and more of those genes for that chemical that you want. So this is a picture. I'm going to actually just do the left side. The right side is just kind of a close-up. But this is called a plasmid. It's inside of a bacteria, and it's that purple loop. And this is like pretend like a human chromosome, a part of a chromosome, human DNA. And we want this protein right here, this one that's in yellow. Let's just say we want more of it. So what we do is we cut it out, and we cut open the plasmid, and we insert that in there. And we actually can then glue it all back together and put it back into the bacteria, as you can see down here. And then when the bacteria multiply, every single one of these bacteria cells will have a copy of that human gene that we want. And here you can kind of just see it kind of like how it might look if you were looking at the bases, but you can literally cut it at certain bases and insert other bases in as long as they'll match up here. And then you've inserted that gene down here into the plasmid. Remember, the purple's the plasmid, the yellow's the gene that we want. So what you're seeing over here is really just a close-up of this right here. And obviously this is called recombinant because you're combining two different organisms, DNA. And there's a couple enzymes and chemicals that you need. You need something called restriction enzymes that they cut the DNA here, and they'll cut it here and here. And then you need something called ligases, L-I-G-A-S-E, and a ligate means to glue together. So the ligases will glue that gene into this plasmid, and then it'll glue everything together. That's called ligase. And I might have said this already, but it involves cutting the DNA in bacteria and in humans, and then they cut from the donor and you put it into the plasmid like a human gene. And like I said, they're specific so that they insert into the plasmid, into the loop, and then there's an enzyme called DNA ligase that pastes the DNA pieces back together, and then you insert it back into the bacteria, and you make multiple copies very quickly, within hours to days. So that's kind of part one of what is this thing we call biotechnology. Now, we're going to get into the uses of it next. Let me just show you where I'm going with that. I'm going to be going to part 2, and I'll do that on a separate video. So I hope this makes sense, and thanks a lot for watching, and we'll see you in the next part.